Hello, everyone, and welcome to today's live broadcast, a sensitive and robust plasmid analysis method by capillary electrophoresis with laser-induced fluorescence detection. Presented by Colleen Reed, Scientist in Analytical Research and Development, Pfizer. I'm Christy Jewell of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by SCIEX. For more information on our sponsor, please visit sciex.com. Now let's get started. I would like to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer questions at the end of the presentation. If you have trouble seeing or hearing our presentation, click on the Support tab or use that Ask a Question box to let us know you're experiencing a problem. This presentation is educational and does offer continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd now like to introduce our speaker, Colleen Reed, Scientist in Analytical Research and Development, Pfizer. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Colleen, you may now begin your presentation.
Thank you very much for the great introduction. Hello, everybody. I'm going to talk about the development of a sensitive and robust plasmid analysis method by capillary electrophoresis, laser-induced fluorescence. So first, I'd like to talk to you about why plasmid is so important for the biopharma industry. Plasmids are used in DNA vaccination, in which naked plasmid DNA is injected directly for expressing protein antigens. Plasmids are also used in CAR T cell therapy as intermediates for making lentiviral vectors, and additionally, plasmids are used in gene therapy as intermediates for making adeno-associated virus vectors, or AAVs. So what is a plasmid? A plasmid is a small DNA molecule within a cell that is physically separated from chromosomal DNA and can replicate independently. Plasmids are most commonly found in bacteria, but are also present in eukaryotic organisms. Artificial or recombinant plasmids are widely used in genetic engineering and the making of viral vectors. Plasmids may be introduced via transformation, as in bacteria, or transfection, as in eukaryotic organisms. So there are three main plasmid isoforms. Those are the supercoiled, or the covalently closed circular, the nicked, or the open circle, and the linear. Here you can see them in the electron microscopy images. We can see that the supercoiled form is highly compact, while the nicked, or open circle isoform is a relaxed circle, and the linear form is just a flat chain. This is a simple schematic diagram of how supercoil form can change into open circle and linear forms. During the processes of plasmid extraction, purification, and storage, it's inevitable that there is some sort of degree of degradation. When a supercoil isoform has a single-stranded cleavage, it becomes open circle form. When there is a double-stranded DNA cleavage, it becomes the linear form. So here's an image of the traditional way that plasmids have been analyzed by the agarose gel electrophoresis. This slide shows data obtained by running a 10 kilobase plasmid sample on a 1% TAE agarose-containing gel with a DNA intercalating dye, ethidium bromide. In lane M, a 1 kilobase plus DNA ladder is loaded. And in lane P2, 200 nanograms of a 10 kilobase plasmid sample was loaded. You can tell that this is a high-quality sample because the major isoform is the supercoil monomer, which runs the fastest and has the highest intensity signal. The linear isoform band runs around the same position as the 10 kilobase marker in the latter. It's barely detectable. The open circle isoform runs slower than the linear isoform, and there's a faint band above the open circle isoform. This is a supercoiled dimer. Now, although agarose gel is very easy to do, it does have its limitation in terms of sensitivity and quantitation. Therefore, that led us to looking into capillary gel electrophoresis as an alternative way for plasmid analysis, especially since we have gained ample experience in using capillary gel electrophoresis for the purity analysis of monoclonal antibodies. So capillary electrophoresis has been extensively used for the separation and analysis of nucleic acids for the past 30 years. This slide shows a simple diagram on how capillary gel electrophoresis works. The capillary, which is an open glass tube with an inner diameter of 30 to 100 microns. The separation occurs when there are charged or polar molecules introduced into a capillary that is filled with a separation gel under the influence of a uniform electric field. The separation gel serves as a molecular sieve for the sample separation. So for this method, we use the SCIEX PA800 Plus instrument and the double-stranded DNA 1000 kit by SCIEX. This kit includes 2 coated DNA capillaries and 3 vials of double-stranded 1000 gel that are lyophilized. The first thing we did in optimizing this plasmid analysis method was to identify the dilution factor for the double-stranded DNA 1000 gel, since this gel was initially optimized and manufactured for the analysis of linear double-stranded DNA. We diluted the double-stranded DNA 1000 gel 5, 7, 10, 15, and 20-fold with 1x TBE buffer. Each diluted gel mixture was mixed with a fluorescent double-stranded DNA intercalating dye, and in this case, we used the LIF Fluor Enhance from SCIEX. A 7 kilobase plasmid was diluted to 8 nanograms per microliter, 1x TBE buffer, and injected by pressure. During the separation at 7.8 kilovolts for 20 minutes, sample peaks were detected by laser-induced fluorescence detection. This slide shows an of the electrophoregrams obtained with the gel at different dilution factors. Better resolution was achieved with the gel dilution factors of 7, 10, and 15, while resolution was not quite as good at the 5x and the 20x solution. So we basically repeated the same experiment, but with one change, one big change. We tried the electrokinetic injection instead of the pressure injection. This slide shows that the best separation was obtained when the double-stranded DNA 1000 gel was diluted tenfold. Therefore, the tenfold dilution was what we used to prepare the separation gel from this point on. So the next parameter we optimized was the sample injection condition. This slide shows results of when a 10 kilobase plasmid was analyzed using the 10x diluted gel with the LIF Fluor Enhanced dye and injected at different pressures for 5 seconds. You can see that when the pressure was reduced from 0.5 psi to 0.1 psi, the resolution of the 2 isoforms were gradually improved. With the 0.1 psi shown in the brown trace, we also did a similar experiment using electrokinetic injection for the sample introduction. In this slide, we used a 10 kilobase plasmid sample that was injected at 2 kilovolts for 4 to 16 seconds. This resolution was around 8 to 12 seconds. We also noticed that the electrokinetic injection gave us greater resolution than the pressure injection. With the gel dilution factor and the sample injection method optimized, we looked into different double-stranded DNA intercalating dyes with the hope of finding one that will offer a higher signal than the LIF Fluor, but then still retain the good resolution between the isoforms. This slide shows the results collected with 4 different dyes in the analysis of a 7 kilobase plasmid sample. You can see both the LIF Fluor and the cyber gold, good resolution and peak shape, while the acidium bromide and the yo-yo dye generated broad peaks. In addition, the cyber gold produced a much higher signal than the LIF Fluor due to its higher quantum yield. Therefore, the cyber gold was used in the final optimized method. So in light of time, I will not go over the details of additional optimization experiments we performed, but I'm happy to share with you the conditions in our optimized method for plasmid analysis. They're listed here. The capillary we used was the coated DNA capillary from CEIX. It was installed with a total length of 40.2 centimeters and an effective length of 30 centimeters. The CEIX double-stranded DNA 1000 gel was diluted with 1x TBE tenfold, and I'd like to point out that also during the optimization of this experiment, we tested the effect of adding a methanol rinse step before routine conditioning method and before routine shutdown method, and we found that this rinse step improved the run life for the DNA capillary. This slide shows the stacked electropherograms obtained by injecting a 10 kilobase plasmid 21 times. You can see that the peak pattern is very consistent between the runs. The main peak is the supercoil monomer peak. The minor peak is the open circle monomer, and this small peak here is the supercoil dimer peak. The results demonstrate excellent repeatability. In this experiment, we used the optimized method to analyze plasmid degradation. A 10 kilobase plasmid sample was stressed at 40 C for 0 to 6. You can see that the supercoil monomer peak gradually becomes smaller, while the open circle peak becomes taller. Therefore, this plasmid analysis method can be used for monitoring plasmid degradation. In this slide, we compared the results obtained with agarose gel electrophoresis with those obtained with capillary gel electrophoresis. The 10 kilobase sample was degraded at 40 C from 0 to 12 weeks. Samples were analyzed by both AGE and CGE. The corrected peak area percent for the supercoiled monomer peak was calculated and plotted in this graph here that you see. Red bars represent results obtained with AGE by staining the gel after the electrophoresis. The blue bars show data obtained with the acidium bromide dye inside the agarose gel. The green bars and the gray bars represent results obtained with CGE, the green being without the methanol rinse, and the gray being with the methanol rinse. The results obtained with the AGE are very consistent with the results of the CGE. Among the two sets of data obtained with AGE, the results collected with the dye inside the agarose gel were more consistent to the CGE results than those collected by staining the gel after the run. There's no significant difference between the results collected without the methanol rinse in CGE and those collected with methanol rinse in the CGE. So in conclusion, we developed a sensitive and robust CE-LIF method for plasmid analysis. We optimized the gel dilution to allow plasmid isoforms to be baseline resolved and quantitated. Electrokinetic injection performed better than pressure injection. Intercalation of different dyes impacted the separation of the isoforms, and Cyber Gold produced the best separation profile for this method. Addition of a methanol rinse step as the first step in the conditioning extended the number of injections per sequence and increased the capillary lifetime to over 100 injections. The topoisoform percents were not affected by the methanol rinse. Experiments with stress samples showed that CE-LIF can quantitate plasmid topoisoforms. So finally, I'd like to acknowledge some of my colleagues here at Pfizer Chesterfield, Yuting Chen, Sneha Chatterjee, and Larry Thompson. And I would also like to thank our great collaborators at SCIEX, Jane Liu, Marcia Santos, and Brandon Bates. Thank you. Okay, let me open up for questions now.
Thank you, Colleen, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, let's get started. Colleen, let's start with this question. Have you analyzed your plasmid samples with HPLC? And if yes, are those results consistent with the CE results?
Yes, using multiple, we've done that using multiple HPLC columns and methods. And the percent open circle tends to be lower, and the supercoiled tends to be higher for the HPLC. The effect tends to be larger as the plasmid size increases. And our data suggests that this is due to low open circle recovery on the LC.
Okay, our next question. I also have a question regarding HPLC analysis of plasmids. Since most plasmid manufacturers use HPLC for plasmid purification, isn't it more convenient to also use HPLC for plasmid quality analysis than using the CE method?
So manufacturers do use column chromatography for plasmid purification, but this is at like the process scale chromatography with beads and pores that are much larger, and unlike the small scale of when we run the ACL or the LC, and that leads to when we run the LC, we have shown the low recovery of the open circle. So CE seems to be more accurate for the quantitation of the topology.
Thank you, Colleen. Our next question. In addition to agarose gel, capillary electrophoresis, and HPLC, did you try any other technique? If yes, how does that technique compare to the agarose gel, CE, and HPLC?
So yes, we have also run AUC, and then so the pre-stained agarose gel, the AUC and the CE method tend to align on both the supercoil and the open circle. The HPLC is really the outlier.
Now, you showed some nice results obtained with plasmids of 7 kb and 10 kb in size by the CE method. Have you tried the same method on plasmids with sizes smaller than 7 kb or bigger than 10 kb?
So we have successfully analyzed plasmids that are higher than 10 kilobases and smaller than 7 kilobases, but you do have to keep in mind that when you're using the CE, that the inner diameter of the DNA capillary is at most 100 microns. So yes, we have varied it, but you definitely have to keep that inner diameter in mind.
Thank you. Now, I have Cyber Green 1 in my lab. Cybergreen 1 binds to double-stranded DNA. Do you think I can use Cybergreen 1 with your CE method?
So Cybergreen 1 would work, but it was not tested. If you were to do that, you would definitely need to optimize the amount of dye used, since the dye is different. And the separation time would likely need to be adjusted too with the different dye. And then also you would have to see if the methanol rinse would be appropriate with using the Cybergreen dye.
Okay, thank you. We have so many great questions coming in, and I want to remind our audience that those questions we do not have time to answer today, and those questions that come in during the on-demand period, they will be addressed by our speaker via the contact information you provided at the time of registration. Okay, our next question is, let's go with, have you tried using uncoated capillary for plasmid analysis?
Yes, but right now for us, that's a work in progress.
Now, Colleen, we have an attendee who understands that cybergold dye is used to label the plasmid DNA with fluorescent signal. They would like to know, does it do anything else?
Well, as we looked at it, it does not seem to affect the topology or the isoforms themselves. And we look at this by comparing it to the AUC and the agarose gel, and since they're all aligned, we do not see that it has an effect on the topology.
And we have time for a few more. I use agarose gel electrophoresis to analyze plasmid isoforms in my lab. It's great to see there are other methods out there for plasmid analysis. Your presentation showed good correlation between the CE data and the agarose gel data. Could you please elaborate a little more on the pros and cons of both agarose gel method and the CE method?
So the agarose gel is inexpensive and it's pretty easy to run. You can run multiple samples at the same time, but that is difficult to automate, like the sample loading. And then also you can find that bands, particularly in the side lanes, like near the edges of the agarose gel, might not be flat. They might kind of have a smile look to them, actually. And this causes issues with the actual quantitation of the bands themselves. And then this problem, this issue with the bands leads to less reproducibility with that method. And then also, if you get a thick band, like something that you might get with a high sample amount in your agarose gel, the migration time tends to go a little bit faster than normal, and this causes variations between the gels as well. So although agarose gel is less sensitive and less quantitative than the CE method, it's a good tool for us to use with our CE data to confirm our work on the CE.
Thank you, Colleen. Now, our final question, what would happen if you don't dilute the dsDNA gel?
Well, so if the double-stranded DNA gel, the undiluted gel basically allows 1,000 base pairs to enter, but so larger molecules with 5,000 base pairs will not be able to enter the gel. So, and so that would affect the plasmids. And so the plasmid size is going to determine this gel dilution, which is required for this separation.
Thank you, Colleen, and thank you for your time today and your important research. Now, I'd also like to thank LabRoots and our sponsor, SCIEX, for underwriting today's educational webcast. One final thank you to our audience for joining us today and for their interesting questions. We have a lot of questions that came in that we were not able to answer, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now, and we hope to see you again soon. Have a great day.